Hello, everyone. Welcome to our workshop. Uh, I'm Max Nomura from Pharmaceuticals Corporation, Japanese chemical and material company, and work as the chair of International Affairs Committee of FARM, Forum for Innovative Regenerative Medicine. It's my great pleasure to have an opportunity to organize this first workshop about the activity for regulatory harmonization in Japanese current situation of regenerative medicine. One of our firm's activity is APACRM. APACRM got started in 2018, is being held as a place to exchange information and opinions on regenerative medicine between Asian countries and regions in order to harmonize regulations on regenerative medicine. The member of APACRM at the moment are these organizations from China, Korea, India, Singapore, Taiwan, and Japan. Today's workshop will focus on prospects and challenges for or against regenerative medicine to promote harmonization and optimization of regulation among Asian countries and regions. Firstly, Mr. Kozo Saeki from Japanese government, Ministry of Economy, Trade and Industry will kindly make opening remarks for the workshop. After that, I will explain a brief introduction of latest APACRM meeting and quick review update on regenerative medicine from Japan, China, Korea, Singapore, India, and Taiwan. Then our colleagues at the firm, Tomo and Hiro, will make presentation about results from APACRM work group 1 for non-clinical assessment of cell therapy product. Okay, moving on opening remarks, Mr. Kozo Saeki have been working as director of Bioindustry Division at METI, Ministry of Economy, Trade and Industry, since July 2021. He is engaged in implementing Japan's industrial policies to promote bioindustry and strengthen vaccine manufacture base against the coronavirus. Prior to this position, he works as senior research scholar at the Edwin D. Reischauer Center for East Asian Studies from 2020 to 2021. Before he was appointed to this position, he served as the executive assistant or special assistant to the Prime Minister Shinzo Abe. He played a role in writing Prime Minister Abe's major policy speeches, including the State of the Union in the Japanese Diet and advisory directory to Prime Minister on industrial policy, trade policy, and energy and climate policy. He worked at the Prime Minister's office from 2013 to 2020, as well as from 2006 to 2009. He also made a contribution to promote semiconductor industry and the lithium-ion battery industry, with strengthening supply chain resilience in electronic industry from 2009 to 2011. He held a master role and diplomacy from Tufts University and the Bachelor of Law at the University of Tokyo. Mr. Saeki, please, opening remarks.
Thank you very much. Thank you very much for a kind introduction, Nomura-san. Thank you very much. Yeah. Yes, I have worked as a speechwriter for the Prime Minister Abe for 8 years, but I'm just focusing on Japanese speeches in Japanese only. So my English is not so much good. I'm sorry for that. First of all, I want to say that. It is my great honor to make the opening remarks of this great workshop today. I'm Kozo Saeki, the Director of Bioindustry Division as a Minister of Economy, Trade, and Industry of Japan. I'm fully engaged in promoting Japanese bioindustry and also recently in enhancing the industries related to the vaccine against COVID-19. As you have already known, new modalities such as the cell and the gene therapy are really changing the world now. They are exactly the key to the treatment of intractable illnesses, the bright future where everyone is living its healthy longevity and where no one, especially no one child, is suffering from the genetic disorder. This kind of future is definitely right there. But in order to realize the commercialization of these regenerative medical products, it is essential to establish a technological base that enables efficient mass production of high quality and highly homogeneous products. It is the living cells with which we are dealing. So the quality of the product is highly dependent upon the manufacturing processes. The outcome differ based on such elements as the culture conditions, used instruments, the methods of freezing the cells, and the way of transportation. In addition, the quality of the raw materials varies greatly depending on the individual donor, the type of tissue, and the method of its collection. Therefore, the standardization of basic manufacturing technologies is extremely important for commercialization of the cell and gene therapy sector today. Japan is determined to play a leading role in this effort. For this purpose, we are trying to establish the new concept of quality by design called QBD, which is an approach to control the manufacturing processes in order to minimize quality variations so that we can improve manufacturing efficiency. The conventional method of quality by testing, the approach to check the quality of each final product, requires great time and cost to obtain a certain amount of qualified products. In order to control quality through QBD, we are analyzing a large number of parameters related to the quality assurance of the final product. For example, temperature, speed of agitation in cultivation, and time in dispensation, and so on. In order to specify these critical quality attributes we call CQA, we are now trying to quantify and to standardize the implicit knowledge in each manufacturing process. This QBD concept will make the industrialized mass production process available for us. One more challenge we are facing is harmonization. These standards should not be confined only to Japan, but should be shared among countries in a form that can be used globally. Many producers suffer from the difference of regulations or data requirements of regulatory bodies. The harmonization of these regulations and practices will make more room for economic growth for the world. In this respect, I would like to express my deep appreciation for the cohesive efforts made by PHARM, the Forum of Innovative Regenerative Medicine in Japan. PHARM has played a leading role in APACRM, Asia Partnership Conference of Regenerative Medicine, for harmonizing and optimizing regulations among Asian countries and this region. we, the government of Japan, will continue to make every effort to encourage such great activities. In conclusion, I would like to express my deepest gratitude to all participants, and I hope that in this workshop, the industry and academia will make a great success. Thank you very much for time. Thank you very much.
Thank you. Thank you for your great opening remarks. It was very encouraging. Exactly, the close communication and discussion about the government, industry, and the academia become the key for everything. We hope to keep good relationship. Thank you.
Thank you.
Okay, next is I will explain the brief introduction of latest APACRM meeting and quick review update on regenerative medicine from Japan, China, Korea, Singapore, India, and Taiwan. As you know, FARM has been established in 2011. Our mission is to accelerate industrialization of regenerative medicine. We have more than 200 member companies now. It consists of pharmaceutical companies, chemicals and material companies, machinery device companies, logistics, insurance, and consulting companies, very diversified. FARM has global partnership by MOU in the field of regenerative medicine industry, including ARM. We think the aging harmonization of regulation is a priority issue. We organize APACRM is to sort out point of difficulty toward harmonization. APACRM got started in 2018, is being held as a place to exchange information and opinion on regenerative medicine between Asian country and region in order to harmonize the regulation of regenerative medicine. The member of APACRM at the moment, the organization from China, Korea, India, Singapore, Taiwan, Japan. To promote harmonization and optimization of the regulation on regenerative medicine products among Asian country and region, it is important to share information and exchange opinion each other. We should build a common view on prospects and challenges for or against regenerative medicine. We believe this activity should maximize the opportunity in the Asian market and promote new products available to Asian people. In turn, that would lead the creation of the market comparable to North America and Europe. This is a brief history of APACRM. On the second APACRM meeting in 2019, we decided to set up the working group activity to discuss more with each expertise. Two focus points were selected on the working level meeting on 2019, and two working group activity have had diligent discussion on each theme since then. Working group 1 is to investigate the differences in non-clinical data package required for cell therapy product in Asian country and region, and to extract to regulatory science issue and limitation. Working group 2 is to discuss about the regulation on starting cells for cellular therapy product in Asian country and region and risk-based approach. Although the third APACRM was called off due to COVID-19 last year, we summarized such deliverable in position paper. One deliverable or worthy one activity has been published in Science Therapy. Output of worthy two activity has already published on regenerative therapy. Recently, fourth APACRM meeting was held on April 2015. Its panel discussion with invited regulatory authority from each country and region were based on those deliverables. In order to disseminate the result of conference into public, we are preparing to publish preceding paper in the international journal. Today's workshop will focus on this worthy activity, that is non-clinical assessment of cell therapy product. We would like to share the result of recent fourth APACRM meeting, its panel discussion with invited regulatory authority from each country and region. Before that, I will make a quick review on Asian update on regenerative medicine from Japan, China, Korea, Singapore, India, and Taiwan. First of all, Japan. Dr. Yoshitsugu Shitaka, from President of Pharm from Japan, provided an update on the cell and gene therapy product development and regulation in Japan. This slide shows comment was in April. Now 6 products was approved in 2020 and 2021, and total number is 13. 9 of these products have been fully approved, while 4 are conditional and time-limited approval. There are the regenerative medicine products approved in Japan now. Despite the pandemic situation, product development have been progressed. 6 products were approved in 2020 and 2021. Currently, 9 of these products have been officially approved, while 4 are conditional and time-limited approval. In addition, marketing application of at least another 2 products have been submitted to the health authority. Overall, the total number is marketed cell and gene therapy product is steadily increasing in Japan. Yescarta and Breyanzi autologous CAR-T product that target CD19, like Kymriah, were approved last December and this March, respectively. These are all CAR-T products for the treatment of severe type of B-cell lymphoma, but with subtle differences in the target indication, cell preparation, and sitting condition. It will be interesting to see how these differences affect their market share. Next is China. Alex, a president of a CMBA from China, shared the current national regulation for regenerative medicine product in China. Pharmaceutical Administration Law of China was amended in 2019. 2 key actions have been taken under the new law. Firstly, the regulator indicated their support toward pharmaceutical innovation aiming to cure human disease. Development of new drugs and therapeutic capable of treating life-threatening disease and rare disease based on the novel mechanism of action, and ameliorating disease by multi-targeting and systemic intervention are encouraged. Secondly, they are trying to strengthen the process control and management of new drug development, clinical trial, and review and approval process. To achieve this, the regulator provides more opportunity of mentoring and consultation to the industry and establish more communication with the industry. Some effort have been done more frequently than before under the newly published regulatory and policy document. Next is Korea. Brian, presenter of CARM and SCRM from Korea, provided the update of product development and regulation in Korea. 16 products have been approved in the market by 2020, and the number of new clinical trials, including gene therapy and gene-modified cells, increasing gradually, and multiple cases of immune cell therapy under development. Most companies in Korea are developing cell therapy, but there are a good number of companies in the gene and gene-modified cell therapy sector, and many Korean companies are doing business overseas recently. The Advanced Regenerative Bio Act was passed by the Congress in August 2019 and enacted 1 year later. The new regulatory process of regenerative medicine consists of 2 tracks. One is an industry-sponsored trial for advanced biopharmaceuticals under the control of Ministry of Food and Drug Safety. The other is for the research trial, regenerative medicine, under the supervision of Ministry of Health and Welfare. Singapore. Casey Wang, a presenter of SAPI from Singapore, presented a current Singapore cell and gene products regulation. The Health Science Authority, HSA, introduced a new category of health product to regulate it under the Health Product Act. Under the law, the cell, tissue, and gene therapeutic product fall under 2 classes, class 1 and class 2 in Singapore. Class 1 is categorized at low risk, for example, minimally manipulated bone marrow or re-implanted for homologous use. Class 2 contain, for example, gene-modified cells, cell grown on scaffold, culture expanded cells, and those of therapeutic gene and genome-based product. For the clinical trial, class 1 are regulated under the human biomedical research for the Ministry of Health. As for class 2, products are required to apply for the clinical trial authorization or submit a clinical trial notification to HSA. India, APARM of April in India presented the regulatory framework and pathways for regenerative medicine in India. Many different guidelines were issued by the regulatory agencies. According to the new drug and clinical trials rule in 2019 issued by the Central Drug Standard Control Organization, CDSCO, any stem cell derived product, genetic therapeutic product, or xenograft intended to be used as a drug will always remain as new drug. Although the final guideline or rule are yet to be released, the definition of stem cell derived product was clarified by CDSCO in 2019. The minimal manipulation of cells cannot be considered as a drug in India. There are 4 approved cell therapy products, and first clinical trial of CAR-T will be initiated in India. According to the National Guideline for Stem Cell Research, which was issued by the Indian Council of Medical Research and Development of Biotechnology in 2017, all stem cell therapy except bone marrow transplant in approved indication need to be conducted as a clinical trial and registered with clinical trial registry from India. Taiwan, Xinzong Lin, presenter of BPIPO from Taiwan, provided development status of regenerative medicine and overview healthcare system in Taiwan. Taiwan adopted a dual track management system of regenerative medicine, which is considered as either medicinal product or medical technology. Under the administrative regulation of specific cellular therapeutic technology, cell therapy technology can be proposed to the Regenerative Medicine Advisory Council and then sent for review by the specific medical technology committee organized by the Ministry of Health and Welfare. Upon approval, the technology can get a conditional 3-year time limited license with renewal subject to regulatory reviews on safety and efficacy. More than 40 hospitals nationwide and 73 items have already been approved for use. Many of these indications are solid cancer. Okay, that is a quick review of Asian update on regenerative medicine from Japan, China, Korea, Singapore, India, and Taiwan. Okay, next is Tom and Hiro will make a presentation about the result from APACRM Working Group 1 for non-clinical assessment of cell therapy product. Tomo-san, please go ahead.
Thank you for giving this opportunity to share APACRM Working Group 1, our Asian activity on this great conference. I am Tomohiro Yoneda, the working group leader on first term of this activity. On behalf of our working group, I'm going to make a presentation about non-clinical assessment of cell therapy product, mainly focusing on mesenchymal stromal cells. We conducted our industry survey and panel discussion based interaction with regulatory agency in Asia. Today, I would like to share their perspective to be compared between countries and regions about non-clinical assessment of cell therapy product. Here is disclaimers. I am an employer in Novartis. So this is today's session outline. At the beginning, I'm going to show background of this activity. Secondly, I will explain our industry survey and panel discussion with regulatory agency in Asia. Then finally, I will suggest future prospect to regulatory harmonization in the medicine field. At first, the background, what is APACRM Working Group 1 and how are our Working Group 1 activity. Apart from low molecular drugs and biologics drugs, regenerative medicine have been growing up as a newly advancement field. So cell therapy is one of the pillars in regenerative medicine field. As Dr. Nomura mentioned that the mission of APACRM is to expedite the launch of regenerative medicine, such as cell and gene therapy, for people in Asia. For reaching out to these missions, it is important to promote harmonization and optimization of the regulation on regenerative medicine product among Asian countries and regions. Here is one of guideline and guidance applicable for human cell therapy product in each Asian country and region. In current situations, there are domestic guidelines and guidance in each country and region. On the other hand, there is no worldwide international guideline. Therefore, the product development of regenerative medicine will be following each domestic guideline and guidance. when the developer develop this in each country and regions. So here is objective of our activities. Our objective is to investigate the difference in non-clinical assessment required for regenerative medicine in Asian country and region based on the domestic regulatory guideline and guidance and the practical experience of each participating members. We would like to extract the regulatory science issue and the limitation and to contribute to the progress of regulatory science in the future. The member in this activity consists of each industry association of regenerative medicines, which are APACRM in India, CMBA in China, BPIPO in Taiwan, SCRM and CARM in Korea, and FIRM firm in Japan. This slide shows contents of our industry survey to sort out the view of Asian country and region necessary for non-clinical evaluation to start clinical study. We created two types of virtual human cell therapy products, mesenchymal stromal cells and induced pluripotent stem cells. We also create the questionnaire to ask the non-clinical assessment to enter clinical study on each Asian country and region. Each member prepare their answer in their own country and regions. After we compare their answer and form the discussion key point, they extract the regulatory science issue and limitations. Here is two virtual human cell therapy products we created. We selected two contrasting cell types, mesenchymal stromal cell and iPS cells. Mesenchymal stromal cells have a long history as a cell source for cell therapy products, and several products have already been marketed in Asia. Therefore, both regulatory agencies and industries have abundant experience and knowledge regarding this cell type. However, on the other hand, the overall development of iPS cell-based cell therapy product is still premature. But each member was curious about their development strategies. That's why we adopt this iPS cell-based product as a virtual cell therapy product item. In the case of the mesenchymal stromal cells-based cell therapy product, we said donor type is allogenic. First in human, adipose tissue-derived cell source, and proliferation is low. So mode of action was defined as immunomodulatory effect via paracrine factors. So rheumatoid arthritis was selected as an indication because there were no approved regenerative medicine product in rheumatoid arthritis. That means this is completely virtual situation, and a validated animal model for rheumatoid arthritis is expected to be available. The dosing regimen in the clinical study was determined based on the chronic nature of disease of rheumatoid arthritis. Incomplete clinical administration route is intra-articular injection. The administration cell number is relatively low, which is available administered to animal model as well. So clinical administration frequency is monthly, repeated dose until 6 times. And genetic manipulation is known. For the iPS cell-based cell therapy product, autologous different edit cell was selected to avoid complexity arising from immune rejection of allogenic cells. First in human, and cell source is human blood cell, proliferation of iPS cell is known to be high. The mode of action is direct supplement effect of progenitor cells via engraftment. Hypothetical hematologic disease caused by the lack of specific functional cells in adult was set as a target engagement. We set systemic dose, high number of cell dose, and single dose. Animal model is known, and genetic manipulation is known. So systemic administration of a high dose regimen is very challenging and has not been attempted in clinical practice. But we set this to get to know our perspective in discussion to explore our future good development strategy. In our survey, we can obtain the fruitful information regarding mesenchymal stroma cell-based product, but obtain in iPS cell-based product due to lack of our experience and specific regulatory guideline and guidance. Therefore, we focus on the mesenchymal stroma cell-based product for future discussions. So this is the questionnaire content. Although we ask more detailed questions one by one, this is high-level summary for that. At first, non-clinical evaluation. So pharmacology, CKBD, safety is needed to start a clinical trial in your country and region. If yes, necessary type of studies are asked, and GLP is needed. For detailed study design, how many animal species or which animal species are the model? How many dosing group, administration route, or dosing frequency are measurement items, and observation period, etc. Based on domestic regulatory guideline and guidance and practical experience of product development, each members prepare the questionnaire answer. So through our survey, we found the similarity and difference in the perspective about non-clinical evaluation in Asian country and region, and extracted the following discussion point with difference. In pharmacology, so the difference were identified for the perspective of proof of concept to develop the rationale supporting therapeutic potential and the exploration to clinical dose setting. In CKBD, collecting tissue sample was one of the major differences. So in general toxicology, study design such as dosing group and dosing frequency and observation period were different across country and region. So in tumorigenicity, the necessity of in vivo study for mesenchymal stroma cells was picked up. Finally, we published our survey result to site therapy as working group 1 deliverable for 2 years. So one of the on the fourth APACRM held on April this year, we conducted the roundtable panel discussion with health authority regulators in Asia, including CDSIO in India, HSA in Singapore, and MFDS in Korea, Taiwan, CDE in Taiwan, and PMDA in Japan. So after the presentation of our working group 1 survey result based on the published article, we have 70 minutes discussion to know the perspective of regulatory agency for non-clinical assessment about thema in center of our batch of mesenchymal stroma cells based product. So next, Let me share the example of industry survey result and the opinion of the panelists from regulatory agency in Asia. So this is perspective regarding number of those group in pharmacology for mesenchymal stromal cell-based virtual cell therapy product. In our working group 1 industry survey, Japan set only one active cell dose group because it is acceptable to show the proof of concept. On the other hand, India, China, Taiwan, and Korea set multiple active cell dose group because an animal study with multiple dose group could gain further insight to predict and understand the pharmacological action in the clinical setting, such as dose response curve, maximum, minimum dose setting. So we asked this point to each panelist from regulatory agency. As a result, Japan regulator has same opinion with Japan industry that study with only one cell dose group is acceptable. Korean regulator is more flexible than Korean industry perspective, and that the use of a multiple dose group is ideal but non-mandatory. If the reliable animal model can be used, or like the rheumatoid arthritis model, one dose group could show satisfactory efficacy. In the case of virtual product of mesenchymal stromal cells, it is possible to use one dose group. So Indian regulator has same opinion with India industry. It is preferred to have 3 dose group based on their domestic guidelines. So this slide shows a study design for general toxicity study that our industry working group indicated. In Japan, one general tox study was prepared. Immunodeficient rat is used, and the route of administration is intra-articular injections, 2 times repeated dose, 1 active dose group, 2 to 4 observation after last dose. So Japan considered that only 1 dose group at maximum tolerated dose or maximum feasible dose is sufficient from the point of hazard risk assessment. The Japanese guidance is that quantitative risk assessment is difficult to apply for cell therapy product because human-derived cell therapy product possibly induce a heterologous immune response in animals, and species difference in the biological activity of each bioactive substance can arise. For example, cytokine. So mesenchymal or stromal cell-based cell therapy product is expected to eliminate at 1 month from body and not accumulate. Therefore, 2 times frequency would be reasonable. According to Japan guidance, around 14 days for the observation period of cell therapy product would be possible because safety assessment of cell therapy would be limited due to immune reactions of the animal study administered human-derived cells. In Korea, 1 tox dose was prepared by using immunodeficient rat, immunoarticular injection, 3 dose group, 6 times repeated dose, followed by 13 weeks observation period after last dose. To understand dose response, multiple doses would be set. Repeated administration according to the clinical protocol, regardless of biodistribution data, would be needed. The standard observation period is 13 weeks, likely after the last dose was set according to Korean guideline. In China, 8 general tox study was prepared. 4 study for for acute tox assessment. Single dose intra-articular and intravenous administration study by using a normal small animal model like rat and large animal model like monkey, respectively, have been utilized in both of their administration route. 3 dosing group, 4-week observation period after last dose would be acceptable. So other 4 studies are 6 times repeated dose intra-articular injection for small and large animal model and intravenous injection for both of animals as well. Observation period of each study is 12 weeks after last dose. So in Taiwan, one general tox study was prepared by using normal rats, intra-articular injection, 3 dosing group, 6 times repeated dose, followed by 4 weeks observation period after last dose. So normal rat is used to observe immune reactions. In India, 11 so many type of study you are prepared. For acute tox assessment, a 6 single dose study consists of intra-articular, intramuscular, and intravenous injection, followed by 2 weeks observations period in normal rabbit and rat, respectively. There are 3 dosing groups. So for subchronic tox assessment, the other 4 studies consist of intramuscular and intravenous injection, followed by a 13 weeks observation period in normal rabbit and rat, respectively. 3 dosing group. So for chronic assessment, only 1 dose, 1 study, 1 dosing group, intravenous, 14 times daily repeated dose, followed by a 2 weeks observation period after last dose in normal rat was planned. For immunogenicity test, only Japan is not considered to conduct the study. So other countries and regions will do the study. In this slide, I'd like to explain one example of panel discussion with health authority regulators regarding general toxicity. For dose frequency, industry survey result is here, as I previously explained. Japan regulator totally agree with Japan industry perspective. Moreover, single dose toxicity study considered if no accumulation of cell is expected in mesenchymal stromal cell-based cell therapy product. Korean regulator have more flexible perspective than our working group survey in Korea. Basically, they admitted to follow the clinical setting, but less than 6 times administration might be considered in such a virtual mesenchymal stromal cell product due to local injection with limited distribution and rapid elimination. Indian regulator consider the Indian industry answer is reasonable based on domestic guideline applicable for small molecule and biologics. The single dose toxicity study should be conducted in 2 rodent species. In this virtual product case, the repeated dose toxicity is required to be conducted. Regarding immunotoxicity assessment for this mesenchymal stromal cell-based cell therapy product, Japan in working group 1 answer no need to do because it is difficult to evaluate human immunogenicity from animals. In addition, mesenchymal stromal cells have been reported to be less immunogenic. On the other hand, other countries and regions need in vivo study, and China and Korea set study as well. Regulator opinion almost aligned with Working Group 1 industry survey. So Japan and Singapore regulators indicated that there is not mandatory to assess immunogenicity for mesenchymal stromal cells. In India, regulator recommended to combine the immunogenicity assessment with general tox study if feasible. A Korean regulator clarified that immunogenicity concern should be addressed for allogenic mesenchymal stromal cells derived cell therapy product. Regarding tumorigenicity, discussion point is necessity of in vivo assessment for mesenchymal stromal cells based product. In Working Group 1 industry survey, only Japan indicated there is no need to conduct in vivo tumorigenicity study for mesenchymal stromal cell based product. If they are produced under GCTP, such as good gene, cellular, and tissue-based product manufacturing practice, because they have been shown to have normal cell proliferation properties beyond a defined culture period. Other country and region need in vivo tumorigenicity study with around 6 months observation period. In line with industry survey, Japan regulator agree with this thought. The perspective of the India regulator is more flexible than industry survey of India. Answer: not mandatory to conduct a tumorigenicity study in every case, and the observation period could be shortened depending on the clinical duration and the nature of the product. The Taiwan regulator and the Korean regulator basically require in vivo tumorigenicity study with 6 months observation period. Although Taiwan regulator recommend to consult with the regulatory agency to determine the study design. So lastly, I'm going to consider future prospect with our suggestion from APACRM Working Group 1. For building the optimal development strategy of regenerative medicine, including cell therapy product, it is important to share regulatory and science information across country and region. For the cultivation toward regulatory harmonization and optimization, we have 4 suggestions. One is communication with regulatory agency. Our approach is to create annual APACRM meeting on April every year to have interaction with regulatory agency in Asia. In addition, Japan industry members have informal casual discussion opportunity with PMDA Japan regulators to share our Working Group 1 activity. The secondary, reporting of the activity, the publication and the presentation at academic meeting, including this MESA conference, would be powerful tool to share our activity to regulatory harmonization into public. Thirdly, the expansion of our activity to other regenerative medicine modality that are cell therapy, such as CAR-T or gene therapy. So later, Dr. Karasawa will explain our second-term activity regarding CAR-T therapy for his later presentations. So lastly, cooperation outside Asia would be important to take worldwide perspective into consideration. We would like to collaborate with ARM on these activities. Therefore, it would be really appreciated if we can reach out to you who are interested in regulatory harmonizations. This is conclusion remark of my presentations. Through the panel discussion with regulatory agency in Asia, we could clarify the difference of the regulation or its underpinning rationale among the participating countries and regions. So sharing the knowledge and correlating data based on the discussion between the industry and the regulatory agency are the first step toward the future harmonization in the regulatory viewpoint about cell therapy in Asian country and region. So this activity is supported by Industry Association of Regenerative Medicine and a variety of members from pharmaceutical and biotech company in Asia. I would like to thank for all of members who are involved in. Thank you for your attention.
I'm Hiroshi Karasawa. I'm playing the leader of second term activity of APACRM Working Group 1. On behalf of APACRM Working Group 1, I will talk about development of regenerative medicine products in Japan and relevant regulation and support from the Japanese regulatory agency, mainly focusing on non-clinical assessment of the product. Here is COI declaration. I'm a scientist employed by Japanese pharmaceutical company Daiichi Sankyo. Here is disclaimer. Here shows contents of my talk. First, I briefly review the regenerative medicine product in Japan. Next, I do point out several difficulties for the development of regenerative medicine products and introduce the support system from the regulatory agency. And finally, I will explain the perspective of Japanese regulatory agency for non-clinical package based on our working group activity in APACRM. This slide shows all the regenerative medicine products approved in Japan in chronological order. Currently, 13 products have been approved and some of which are approved as conditional and time-limited approval. The first one is JACE in 2007. It's an autologous keratinocyte sheet product used for severe burns. After that, many types of products are approved. Several types of cell sheet products, bone marrow-derived MSCs, and CD19 CAR-T products. There are 3 products. And recently, gene therapy products, including AAV, plasmid, and oncolytic virus were approved. It is of note that many of these products have an origin outside Japan, mostly in the US. These are under the several types of international collaboration scheme, ranging from licensing in from the bioventure to the development by Japanese subsidiary company of Mega Pharma. These activities help Japanese patients suffering from the incurable and life-threatening diseases, and we hope more US companies have interest in delivering their novel and effective products to Japanese patients. Here, I'd like to consider the difficulties in the development of regenerative medicine products, particularly keeping in mind the distinction from the conventional medicines. First, in quality control, several issues associated with biologically derived raw materials are pointed. One of the which is the difficulty of sterilization of the raw material and the product in case of the regenerative medicines. In non-clinical assessment, the point is also the one which is inevitable as regenerative medicine products. Obviously, it is intrinsically difficult to evaluate the human cells and tissues in animals. Furthermore, there exists the technical issue and limitation of cellular kinetics and biodistribution study, because the target is not the molecule, but the living cell. This is linked to the clinical trial in which exploration of animal cell dose to human is a big challenge, because the molecule-based PKPD strategy cannot be applied in regenerative medicine products, except for the CAR-T or some products. Another difficulties in the clinical trial may come from the indication of the product. In many cases, the targeted diseases of the regenerative medicine products are very severe or need special settings to perform. Thus, the number of patients who can be enrolled in the study is limited. Among these issues here, I mainly focuses on the non-clinical assessment and some of issues in the clinical trial in the following slides. From here, using some slides, I will explain the systems supporting the development of the regenerative medicine products in Japan by a Japanese regulatory agency, PMDA. Currently, the scheme is under the law named the Pharmaceuticals, Medical Devices, and the Other Therapeutic Products Act, PMD Act. This is a revised version of the former Pharmaceutical Affairs Act that was covering only the drugs and the medical devices. The PMD Act since 2014 have covered regenerative medicine products and established the scheme supporting and accelerating the development of the regenerative medicine products. These three are most important items. The first one is a regulatory science strategy consultation with PMDA, which has particular support for the academia or venture company. The second one is Sakigake designation. Sakigake means forerunner in Japanese, and it is a scheme aiming at early practical application for innovative medicines, including regenerative medicine. The third one, conditional and time-limited approval, is an expedient approval systems. In the following slides, details of each scheme will be shown. This slide shows one of the important aims of regulatory science strategy consultation for regenerative medicine products. As you know, between the cutting-edge basic research and the practical use of the technology, a valley of death is lying. This is a significant barrier for the development by academia or venture company with limited experience in developing the product. PMDA offers the opportunity to intimate communication between applicant and PMDA. Consultation covers all steps in the development to address the difficulties in the development of the regenerative medicine, as mentioned in the previous slides. Particularly for quality and non-clinical safety studies, specific opportunities of the consultation is offered. These will help to design the optimal developmental strategy. This slide shows the scheme of the Sakigake designation. It resembles the breakthrough therapy and fast track designation in the United States, and mainly consists of the rolling review and 6-month review. Major criteria for the designation of Sakigake are shown here. The product must be for diseases in dire need of innovative therapy and not restricted to the regenerative medicines. It deals with the application for approval firstly in Japan or simultaneously first in case of the global development. It requires the expected prominent effectiveness in the clinical setting, thus the dataset of non-clinical pharmacology and the design of the phase 1 study is very important to elucidate the prominent effectiveness. Currently, 2 regenerative medicine products. One is an autologous BMSC product for the spinal cord injury, and another is oncolytic virus for malignant glioma, have been approved through the Sakigake scheme in Japan. The last item is the conditional and time-limited approval. This scheme greatly shortened the time for the marketing of the product. In the case in which conducting the control study with sufficient number of patients is difficult because of the limited number of patients, or time needed to complete study is too long, this scheme is applied for delivering the potential new therapy for the patients suffering with devastating disease with virtually no effective therapy. For this purpose, PMDA grants conditional approval based on the results of the early phase study. For this point, most important factor is the confirmation of the safety of the product. For efficacy, it is sufficient if the effectiveness is expected with limited number of patients. This scheme is a matter of discussion as it has a potential of reimbursing less effective new therapy. Thus, it is very important to collecting sufficient data within the 7-year post-marketing period to judge the full marketing authorization or revocation. It should be understood that the PMDA is taking the risk of transiently overestimating the product, considering for the benefit of patients suffering from the devastating diseases. Currently, 4 regenerative medicine products have been approved and under post-marketing confirmation phase. From this slide, I'd like to change the topic to our findings in working group activity about the non-clinical assessment. As the previous speaker, Tomo, presented, we compared the non-clinical package required from the regulatory agency among 5 country regions in Asia by setting the MSC-based virtual product shown here and reviewing the package from multiple viewpoints like these. Through this activity, we found similarities and differences in the perspective about non-clinical evaluation in each country. In the following slides, I'd like to show the perspective characteristic to Japan, particularly focusing the position and interaction of the regulatory agency and industry. Non-clinical test package prepared by Japan team was most concise among 5 countries regions, and PMDA basically agreed with the view of the industry. Of course, the test packages prepared by other teams are made referring the guidance of each country, but some difference were noted between industry and regulatory of each country. The possible reason of the coincidence of Japan industry and regulatory are shown here. One is that PMDA has already organized and addressed the issues specific to the non-clinical studies of regenerative medicine products. This includes the topics such as how to show the POC in animals with human cells, and how to assess the risk hazard in animals with human cells, how to assess the tumorigenicity of MSC in vivo. The second point is that these concepts of PMDA have been shared with industry through the guideline and guidance published by PMDA. As an example of that, I show one of the basic views of Japan to non-clinical evaluation of regenerative medicine products. That is the limitation of evaluating human cells in animals. This view was most typically reflected to dose group setting in our working group activity, in which Japan team set only one dose group, while others set multiple dose groups. Here shows the rationale for that in pharmacology and safety studies, respectively. For pharmacology evaluation, aim of the study is to demonstrate the effectiveness of the product. In other words, showing the proof of concept. Thus, one dose group with optimal cell dose is sufficient in most cases. For safety evaluation, aim of the study is to identify potential hazard. Quantitative risk assessment is inapplicable to cell therapy products. Thus, one dose group with maximum tolerated dose or maximum feasible dose is sufficient in most cases. These ideas are shared by industry and regulatory in Japan. The next example is the views on tumorigenicity assessment. For this topic, the view of PMDA have been shared via guideline issued on 2019. For ESC and iPSC-derived products, in vivo tumorigenicity study to assess the tumorigenic potentials of the final product in clinical planned administration site is mandatory. For MSC-derived products, in vivo tumorigenicity study is not necessary as far as the product is produced under GCTP and in vitro analyses show no abnormality in their growth profile. Based on this view, in our working group activity, Japan team did not set the in vivo tumorigenicity study as a safety study package. PMDA emphasizes on the importance of understanding the profiles and characteristics of each product and the flexible attitude based on the scientific rationale. Other than shown in the basic views like above, PMDA offers the regulatory science strategy consultation for the safety assessment of regenerative medicine products to address the issues on a case-by-case basis. Okay, in the last few slides, I briefly introduce current activity of our working group. As an activity of the second term, we are focusing on the non-clinical assessment of CAR-T products. The objective is to investigate the non-clinical assessment for CAR-T in Asia country regions for the future harmonization, and 32 professionals from these 5 countries regions are involved. We have 2 aims. And in aim 1, we do case study again to compare the non-clinical data package required for IND submission using the virtual CAR-T products with a conventional profile. This is a graphical summary of the conventional virtual CAR-T products. It is set as an autologous CAR-T product targeting the virtual target molecule CDXX expressed in B-cell malignancy cell. The basic profiles including the detailed design of CAR-T cells and profiles of target cells here, malignant B cells, and also normal B cells expressing CDXX. And basic manufacturing and administration process are defined. These are similar to those of the existing CD19 CAR-T products, and we are planning to compare the basic views for CAR-T products among Asian countries using this conventional CAR-T. In addition, in aim 2, we will find and discuss the non-clinical assessment needed for the specific component of advanced CAR-T products. For example, novel binder, armoring technology, allogeneic off-the-shelf CAR-T, or other cell type than T-cell, etc. In the end, I briefly introduce current outcome of Working Group 1 activity. Before starting the case study with a virtual CAR-T product, we did comparison of non-clinical packages of 3 CAR-T products, Kymriah, Yescarta, and Breyange, all approved in Japan. And we found that these 3 products achieved approval by independent strategy by supporting the efficacy and safety of the product using different data set of in vivo and in vitro studies. For example, Yescarta did not conduct in vivo pharmacology study with human CAR-T cells because of GVHD reaction. Instead, it did in vivo study with mouse CAR-T cells. Similarly, in safety study, Kymriah alone did in vivo general tox study, while others did not because of GVHD reaction. This may suggest again the flexibility of PMDA to evaluate the data set based on the different rationale. And finally, I'd like to say, as a concluding remark of this session, it is very important for industry to closely communicate with regulatory agency to clearly show the rationale of the applicant in the development of regenerative medicine products. This is the end of my presentation, and I'm happy if you have interest in developing your products in Japan and in Asia. Thank you. Finally, I really appreciate all the members involved in APACRM Working Group 1 and other persons and organizations, including firm and regulatory agencies, for generous support for our activity. Thank you very much.
Okay. Thank you for your all and the presentation. This part is the quick question for maybe Hiro regarding next virtual CAR-T activity. What do you think? What's the main hurdle do you think at the moment?
Thank you, Max. Thank you for your question. Yes, for the comparison of the non-clinical package for conventional CAR-T, I think it is not so difficult because we have an experience in the term 1 for the comparison of the non-clinical package of the MSC or iPSC. So I think we can, and also there are some products already approved in US, EU, and in Japan. So we can, each country can prepare the good package based on their regulatory rules. So the M1 is not so difficult, but for M2, for the discussion and for discussion of the non-clinical assessment needed for the special components of the advanced CAR-T product, because the advanced CAR-T is composed of the novel technology. So we should know its technology very well and then catch the latest information about that. And also the regulatory agency have no experience in dealing with such new technology and new materials. So yeah, that's a big challenge for us. And for that, it is very helpful to, if we can communicate with the regulatory agency in each country, that will help for our activity and future harmonization.
Mm-hmm. Okay, thank you. Okay, next is the quick question to Tomo about future prospect. What do you think? What action should we do next?
Yeah, actually, so thank you for having a question, Max. Actually, as I presented, I make my presentations. So our objective of the APACRM is for regulatory harmonization and optimization. And today we're going to focus on the Asian perspective, including the China, Taiwan, Korea, India, and Japan. So that's why, so the other Southwest Asia, for example, or the Thailand, or some kind of Malaysia, or some kind of country, we pay attention to their regulation and be involved in their perspective. On the other hand, so for the regulatory harmonization and optimization, it is important to focus on not only Asian perspective, but also the worldwide perspective. And actually, so in the advancement of the regenerative medicine, where we, how can I say, the promoting in the Europe and Americans at first. That's why we need to ask the collaboration with the Industrial Association of Regenerative Medicine in Asia, in America, or USA, or EU. So that's why, so it would be helpful for us to collaborate with the ARM about this activity toward the cultivation of regulatory harmonization and harmonization and optimizations.
Thank you. Good answer. Okay, maybe almost time to close. So thank you for all participants today. So appreciate it. This workshop focused on the one result of APACRM meeting, non-clinical assessment on the cell therapy product. I think the environment of harmonization will getting better more by sharing experience with product and development opinion and about the regulatory guideline in each country and region. So Pharm will continue to address way to foster a better environment for regenerative medicine with the health authorities and government, like academia, and the industry. Okay, that's all. So thank you for your participation. Thank you very much. Bye.
Thank you so much.
Thank you so much.